Hi, hello, good morning everyone. I'm Dr. Harika Kishore. On behalf of Vistas Learning, I'm here as your biology educator. Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, pre-recorded sessions, study materials, animated videos, and also, moreover, you're going to get this lecture notes in the form of PDFs. Get your acquaintance with all the channels available and subscribe to the channel Vistas Learning Class 11 and 12. If you find this session useful and interesting, please click on the like as well as share this session with your friends. And also click on the bell icon such that you are going to get all the upcoming video notifications. Right, now let's start our today's session where we have been discussing about the chapter that is molecular basis of inheritance, which comes under the unit genetics and evolution. So in the previous sessions, we have been discussing about different important processes of molecular biology like replication, transcription and translation. After that, we need to understand about this gene expression can regulate what we call it as a regulation of gene expression. A gene, if you consider, which is present in a cell, gene, which is sequence of DNA, and this sequence of DNA express which leads to the formation of proteins. That expression of each and every gene happens by means of first transcription. It transcribes into mRNA and further this mRNA will translate to produce polypeptide chain and that is your protein. This is translation. So here expression of genes results ultimately in the formation of proteins. And these proteins will perform a particular functions or set of functions. So here, regulation of gene expression refers to as a very broad term because it can be regulated at different levels. That maybe occurs at various levels. If you consider prokaryotic cells and eukaryotic cells, this regulation will be it differs in different levels. If you come to the prokaryotic cell, it happens predominantly towards the initial stages of the transcription, that is transcription initiation. If you move on to the eukaryotic, where the higher organization can be seen, where this regulation can be done at different levels, like transcription, and we well know that the transcription results in the formation of hnRNA, and that is what heterogeneous nuclear RNA or otherwise called as primary transcript. And this primary transcript will further undergo post-transcriptional modification where we see like tailing, capping, splicing happens. During this stage also regulation can be happened. And once the processed mRNA is produced out of the modifications, that is post-transcriptional modification, this mature mRNA or processed mRNA will move from the nucleus to reach to the cytoplasm. While its movement, that is transferring from the nucleus to the cytoplasm also, there is a checkpoint where it can regulate, that is the gene expression can be regulated. And once it successfully reaches nucleus, where the further process, that is translation, happens, and even at the level of the translation also regulation is happening in the case of eukaryotic cells. So considering that gene expression results in the formation of polypeptide is nothing but protein. Here is a gene which expresses ultimately towards the production of the protein. The ultimate expression of the gene is a protein synthesis, and for that protein synthesis to be happen, first the gene has to transcribe itself into mRNA, and that transcribed mRNA has to translate further into protein. So this is the ultimate expression of the genes. So considering that gene expression results in the formation of polypeptide, and that polypeptide is nothing but protein. it can be regulated, that production, synthesis of the protein can be regulated at several levels. Let's look at that several levels at which the gene expression can be regulated. So here, if you consider this as a cell, eukaryotic cell, nucleus, a well-developed nucleus is present in the eukaryotic cell, where we have DNA, which transcribes, transcription happens here, where there is a copying down of the genetic information which is present in the DNA to RNA. So this is DNA and this is hnRNA because it is a primary transcript. Then this will undergo further modification, what we consider as a post-transcriptional modification, and it will now produce processed mRNA. This is the primary transcript and this is the processed or mature mRNA. Once the mature mRNA is produced inside the nucleus, this is cytoplasm. Now it will transport from the nucleus to the cytoplasm. So we get this processed mRNA in the cytoplasm. Now in the cytoplasm, further translation happens, finally leads to the production or synthesis of polypeptide chain, that is protein. So during this process, altogether we can call it as a gene expression. The gene is expressing in terms of the production of the protein, and it can be regulated at various levels. Like initially it can be regulated at transcription, and it can be regulated during post-transcriptional modification, that is a splicing, where capping, tailing happens, which converts the hnRNA into processed mRNA. And even the mRNA is also produced, then also it can regulate while the transport of this mRNA from nucleus to the cytoplasm is happening. Once the mRNA reaches the cytoplasm successfully, then also regulation can be happened at the level of translation, where the mRNA is finally going to translate to produce a sequence of the amino acids, and that is called as polypeptide chain protein. So here, these are all the different levels at which the regulation of the gene expression is possible in terms of eukaryotes. So here, in eukaryotes, the regulation could be exerted at different levels, various levels, starting with transcriptional level. See, transcription, where DNA is resulted in the formation of hnRNA, and that hnRNA is the primary transcript. This primary transcript is nothing but hnRNA, heterogeneous nuclear RNA. Now, even hnRNA is produced, then also it can be regulated at a second level, and that is called as processing level, post-transcriptional modification, that is regulation of splicing. Splicing is nothing but where the non-coding genes are being removed and attached with the coding sequences. That means introns are removed out and exons are joined to form a mature mRNA. Now, the further level of regulation can be seen at transport of mRNA from nucleus to cytoplasm, from nucleus to the cytoplasm, and this is a third check. So this is at the level of transcription, at the level of post-transcriptional modification, that is the process of splicing, and at the level of the transport of the mRNA from the nucleus to the cytoplasm, and finally it can be takes place at the level of translation, that is a production of proteins. So mRNA has been transported. Now we get this mRNA in the cytoplasm. Even after this, translation can be regulated. So these are the different levels: transcription, post-transcriptional modification, transport of the mRNA from the nucleus to the cytoplasm, and finally at the level of translation. This is all possible in terms of eukaryotic organisms. Then what is about prokaryotes? As we already said, prokaryotes do not show the separate compartmentalization where the transcription is going to happen in the nucleus and the translation is going to happen in the cytoplasm. The same way as we seen in the case of eukaryotes, this compartmentation, that is a differentiation of the site where the transcription is going to happen, where the translation is going to happen. This is not present in the case of prokaryotes. Then what do we find in the prokaryotes? See, in the prokaryotes, the check is towards the initiation of the transcription. So, if you look at the cell of the bacteria, Escherichia coli, a prokaryotic cell, there is no differentiation of a well-defined nucleus is not present in the prokaryotic cells. So, transcription as well as translation, both the processes are coupled together. That means before the actual transcription completes, translation starts already. So, that is what we can consider the process, like transcription and translation are coupled together, and that processing together, happening together can be seen in the case of prokaryotic organisms. So, in prokaryotes, control of the rate of transcriptional initiation of the transcription itself is the level predominant site for the control of the gene expression. But in eukaryotes, we have a different several levels to which this gene expression can be regulated at transcription level, at post-transcriptional modification level, at transport of the mRNA from the nucleus to the cytoplasm level, and even towards the process of translation. Now, so here, the gene in a cell, so this is a cell, a gene, sequence of DNA, are expressed to perform a particular function. Yes. See, ultimately, each gene will first transcribe and then to translate to produce protein. And this produced protein will function as an enzyme, or it will function as a hormone, or it may function as a receptors. Finally, this protein will show functions. It will show biochemical responses, and this will show single function or a set of functions. So, particular function, a single function or a set of the functions. That means we can say the gene which is present in the cell shows its function through its expression, and this expression happens by means of first transcription and then translate into protein. The produced protein will show the function or set of the function. It may act as a single enzyme, or it may act depending upon the need that is present in a particular cell. It is going to show its function or set of functions. For example, to make you understand this, if an enzyme called beta galactosidase, if the produced protein, translated protein, need to function as an enzyme, what happened here? For example, that enzyme is beta galactosidase, is synthesized in Escherichia coli. It is used to catalyze the hydrolysis of disaccharide. What is the function of beta galactosidase? Beta galactosidase. This beta galactosidase is going to act on lactose it is going to show hydrolysis reaction such that disaccharide, which is made up of two monosaccharide units, are going to separate to produce individual monomers, that is monosaccharide units. What are that monosaccharide units? One is glucose plus and other one is galactose. One is glucose and galactose. See, lactose, it is a disaccharide which undergoes hydrolysis to produce two monosaccharide units, and that monosaccharides are glucose and galactose. So lactose breakdowns to produce glucose and galactose, and this reaction is catalyzed by an enzyme, and that enzyme is beta galactosidase. So here, for example, if an enzyme called beta galactosidase is synthesized in the Escherichia coli, which is used to catalyze the hydrolysis of a disaccharide, and that disaccharide means which is made up of two monosaccharide units, which is lactose into galactose and glucose. The bacteria use them as a source of energy, and this produced glucose will be acts as a source of the energy for the bacteria to grow. So here, depending upon the need, see, in a medium, bacteria is growing and it requires glucose from lactose. That means the lactose has to break down to produce galactose and glucose. In that way, it needs enzyme, and that enzyme is coming from protein. So depending upon the need, depending upon the function to be performed, the gene expression will take place. Now, so here, hence if the bacteria do not have the lactose, for example, see, consider this is a petri dish, petri plate, where the growth of the bacteria is present. And this petri plate is having a medium where we have given glucose. Just now we said that lactose is a disaccharide which will undergo hydrolysis in the presence of beta galactosidase to produce two monomeric units, that is monosaccharides, and that are galactose plus glucose. Consider first example. In this example, let's consider the petri dish is provided with a medium, and that medium is having glucose. If the bacteria is directly having glucose, then there is no need for the bacteria to break down the lactose into galactose and glucose, and thereby using this glucose as a source of energy. Already energy source has been directly provided in the form of glucose itself. Then there is no need for the bacteria to express the gene which code for beta galactosidase, right? So it has to regulate this gene expression because there is no need for the lactose to be broken down into glucose and galactose, and that glucose to be used as a source of energy, as the petri dish is provided with a medium, and that medium is already having glucose directly itself. So in the second example, let's take no lactose is available for the bacteria. There is no lactose at all. In that case also, if no lactose is available, there is no need for the beta galactosidase. Beta galactosidase functions on the lactose. If there is no lactose, there is no need for the requirement for the beta galactosidase enzyme. In that case also, we can regulate, it can stop the expression of the gene which code for the beta galactosidase, right? Hence, if the bacteria do not have lactose, there is no lactose available around the bacteria to be utilized for the source of energy. They would no longer require the synthesis of the enzyme beta galactosidase. That is what we discussed in the second example, where there is no lactose available in the medium. If there is no lactose available, there is no need for the beta galactosidase to be produced, to be expressed by the genes. Therefore, in simple terms, we can say that it is a metabolic, physiological and environment conditions that regulate the expression of the genes. If there is a need for that particular enzyme or for that particular protein to be produced, then only the gene will express. or else it has to be regulated. It has to be stopped in order to stop the production of the required, that is whatever the protein that is producing. If there is a need, that protein has to be produced. If there is no need, that protein should be regulated. That is the point. That is the importance of regulation of gene expression. Now, here, the development and the differentiation of the embryo to the infant, and thereby the infant is going to develop into an adult. All this is a coordinated and well orderly going fashion. So that is what we consider this as a gene expression. See, the development and the differentiation of the embryo into adult organisms are always a result of well coordinated. It has to be coordinated, well coordinated regulation of expression at several sets of genes. See, differentiation of the embryo into adult requires the regulation at a very different sets of, that is several sets of genes. Now, okay, here in a transcription unit, while we are discussing about the transcription process, we mentioned about the transcription unit. Transcription unit where we'll have promoter, structural genes, terminator, right? In a transcription unit, the activity of enzyme RNA polymerizes at a given promoter. We know that the enzyme RNA polymerizes will identify promoter, then after it is able to transcribe this DNA into mRNA. Here, in a given promoter, this is the promoter region of the transcription unit. It is in turn regulated by interaction with accessory proteins. They are regulatory, set of regulatory accessory proteins which affects its ability to recognize start site. This is the start site, which this RNA polymerase's ability to recognize the promoter is under the control of the accessory proteins, that is, it is going to regulate. That is what we call it as a regulatory proteins. These regulatory proteins, again of two types. See, RNA polymerase is needed to transcribe in order to produce the mRNA. First, this RNA polymerase has to recognize the promoter, which is a starting site, start site. Before that, it is under the control. This is under the control of regulatory proteins. And here the point is, this regulatory proteins are of two types. It can regulate positively by acting as activators, or it can regulate negatively by acting as repressors. So, the regulatory proteins can act both positively as well as negatively. The positive action is shown by the activators, and the negative action is shown by the production of the repressors. Repressors are nothing but the regulatory protein. If they regulate negatively, we call it as repressors. Now, so here, the accessibility of the promoter region of a prokaryotic DNA is in many cases regulated by the interaction of proteins, and that proteins with the sequences we termed it as a operator. We do find the presence of operators before the structural genes in the case of prokaryotic organisms, DNA. So this operator to which the regulatory protein, whether the regulatory protein will be an activator or repressor, will go into and bind to the operator. And here there is a specific operator to be present in each operon, and for that specific operator, it will have specific repressor proteins. So, the operator region is adjacent to the promoter element. Here is a promoter. To its adjacent, we find the operator in the operons. In most of the operons, and in most cases, the sequence of the operators binds to the repressor proteins, and these repressor proteins will regulate negatively. If it regulate positively, we call it as an activators, right? Now, so here, As I already said that. See, we have operator which is present adjacent to the promoter. Each and every operon will have a specific operator which will have a specific repressor proteins. These repressor proteins are going to regulate negatively. So, each operon has its own specific operator. Each operon has its own specific operators. Operator will be present next to the promoter element and specific receptors. These receptors are produced by the I genes. Now, for example, to understand how the repressors are going to act as a regulatory proteins are expressed to produce these repressors, and how these repressors are going to stop or not going to stop. Let's see. For example, let's take an operon, and that operon is lac operon. Lac for lactose. Lac operon. In lac operon, as we just now said that each and every operon, in this example we are taking it as a lac operon. That means lac operon has its specific operator, that is lac operator, and that specific operator will have its specific repressors, specific lac repressors. So, lac operon is present only in the lac operators will present only in the lac operon, and in turn it interacts specially with the lac repressors only. So, each operon will have its specific operator, each operator will have its specific repressors. If it is lac operon in this example, this lac operon will have its specific lac operator gene, and that lac operator will have its specific lac repressor gene. Now, repressor protein. So, this is how we can see next to the promoter will have operator. And before that, we have the regulatory gene, which expression will produce, whether it may produce activator or repressor. And here, in this case, it is producing repressor mRNA. And now this transcribed repressor mRNA is going to translate to give stress to the protein, and that protein is called as repressor. And this repressor is going to bind to the operator because it is going to induce negatively to regulate the expression of lactose. Now, here, the elucidation, understanding, explanation of the lac operon was also a result of close association between a geneticist and a biochemist. Both scientists worked together to understand, to make us understand, to give the concept clear about the lac operon, and that scientists are Francois Jacob and Jacques Monod, popularly called as Jacob-Monod series. Jacob-Monod, these are the pair of the scientists who worked hard to explain the operon concept, who worked hard to explain lac operon. So, they are first to made you understand, explain, made this difficult concept to understand easily, to elucidate a transcriptionally regulated system. Transcriptionally, see, there are so many levels. And if it comes to the prokaryotic cells, example Escherichia coli, the regulation will be happened at the transcription level. That is the initiation of the transcription, and that is what we call this regulated system as a transcriptionally regulated system. There are so many systems like translationally regulated system or post-transcriptional modification regulatory system, as these are the cells which belongs to the prokaryotic cells. In prokaryotic cell, the regulation happens at the transcriptional level. That is what we consider it as a transcriptionally regulated system, right? So, this is an image of the lac operon. Coming up slides, we are going to deal in depth about this regulation, how the repressor are going to bind to the operator in order to stop the transcription of the mRNA, and further the production of the enzymes are going to be stopped. Now, so here, just now we mentioned lac operon. Lac means lactose. It is a polycystronic structural gene We well know that the monocystronic genes are present in the eukaryotic cells, whereas in the prokaryotic cells, we find the presence of the polycystronic structural genes. These polycystronic structural genes are going to produce several proteins. Here, it is going to regulate by a common promoter. If the set of the genes are undergoing regulation by the presence of, under the control of a single promoter, then that we consider it as a operon. And then that type of arrangement where cluster of the genes, group of the genes are under the control of a single promoter, and that is called as operon. So here common promoter as well as regulatory genes. These regulatory genes are going to produce repressors here. Such arrangement is very common in bacteria, where in bacteria, prokaryotic cells, we do find the presence of the polycystronic genes. Polycystronic genes means which will express to produce several proteins. Poly means many. Many proteins are produced by the expression of this such genes. And is refers to as operon. Operon is a cluster of the genes where the regulation expression of this cluster of genes are regulated by the presence of a single promoter and a regulatory genes. Now, so this is how the regulation happens. Just now we said that we have a cluster of genes, and that cluster of structural genes which are present in the lac operon or ZYA. So look at here. Not only lac operon, there are other set of the operons also. Like, to name few, lac operon, tryptophan operon, arabinose operon, histidine operon, valine operon. Let's understand about the lac operon first. So, lac operon consists of regulatory gene. If you look at the lac operon, we find the presence of, this is the promoter for this regulatory gene. We find the presence of regulatory gene, and that regulatory gene is expressed by I gene. Where do you get the letter I? The I gene, here the term I doesn't refers to inducer, rather it is derived from the word inhibitor, because the lac operon is negatively induced by the presence of the repressor, and this repressor is going to be inhibit. That is what this I gene, which is going to transcript into repressor mRNA, and further this repressor mRNA are going to translate into repressor, and this repressor is going to bind to the operator which is present next to the promoter. As it is inhibiting, we call it as an I gene. Inhibition, I gene, right? And the three structural gene in the lac operon, not only we find the presence of the I gene, but also we have the presence of the structural genes. How many structural genes? 3 structural genes. What are they? Z, Y, A. Okay, let's look at into the function of this Z, Y, A structural genes. Now, here is the promoter, I gene, promoter, operator. These are the structural genes. From here to here, we have structural gene. What are they? One is Z gene, Y gene, A gene, right? Now, the I gene codes for the repressor. It is a protein, repressor of the lac operon. And the Z gene codes for the beta galactosidase. By now, we know the function of beta galactosidase. Beta galactosidase is an enzyme which acts as a catalyst, and it can catalyze the lactose hydrolysis into galactose plus glucose. This reaction is catalyzed by an enzyme, and that is called as beta galactosidase. Beta galactosidase is the enzyme which is coming from the gene, and that gene is called as Z gene. The Z gene code for the beta galactosidase, in short we can call it as a beta gal, which is primarily responsible for the hydrolysis of disaccharide, that is lactose, into its monomeric units. One is galactose and other one is glucose, and this glucose will act as a source of energy for the bacterium, right? Now, next we have Z, we have Y, we have A. Now we have known that Z code for the beta galactosidase, which helps in the catalysis of the hydrolysis, that is a breakdown of the lactose into galactose and glucose. Now, what is the function of next structural gene, that is Y gene? Y gene codes for an other enzyme and that is called as permease enzyme. What is the function of this permease enzyme? It will increase the permeability of the cell to the enzyme beta galactosidase. This beta galactosidase has to enter into the cell such that it can work on the lactose, make the breakdown of the lactose into galactose and glucose. To proceed with this action, that is to proceed with this reaction, the beta galactosidase has to get its entry into the cell, and that permeability of the cell towards the beta galactosidase is increased by the presence of an enzyme, and that is called as permeases. Permeases gives the permeability of the cell for beta galactosidase. Now, the final structural gene, that is A gene, it will code for another enzyme, and that enzyme is trans-acetylase, which is useful for the transfer of the acetyl group. Now, hence all the three genes Z, Y, A, beta galactosidase and permeases, finally trans-acetylases. These gene products are nothing but enzymes. In operon are required, all the three gene products are required for the metabolism of lactose, Therefore, in most other operons as well, not only in the operons, even in the other operons as well, the genes present in the operons needed together to function in and coordinated, that is same or related, that has to be show its function in the same or related metabolic activity, that is pathway. Now, lactose is a substrate for the enzyme beta galactosidase, right? If you consider this is a reaction, lactose in the presence of an enzyme beta galactosidase, it is going to break down into galactose plus glucose. Here, if you consider this is a reaction where lactose is a substrate, beta galactosidase is an enzyme, galactose and glucose are the products. Here, the substrate, that is a lactose. Lactose is a substrate for the enzyme beta galactosidase, and it regulates switching on and off of the operon, because it is going to act as lactose is going to act as an inducer. See, when we find the requirement for the presence of beta galactosidase, if lactose is present, in the starting of this session only we discussed that the presence of the lactose decides whether the beta galactosidase has to come or not. Simple. If lactose is present, it required beta galactosidase. So, the expression of the gene which codes for the beta galactosidase is needed. If the lactose is not there, then why do we need the presence of the beta galactosidase? We don't need the beta galactosidase to be present, because it need to work on the lactose. If lactose is not there, there is no work for the beta galactosidase itself. So, hence it is termed as a inducer. Here, that inducer is nothing but lactose. If the lactose is present, it is going to induce the expression of the gene which codes for the beta galactosidase. And in the absence of the preferred carbon sources such as glucose, if lactose is provided in the growth of the medium of bacteria, the lactose is transported into the cell through the action of the permeases. See, for example, this is a petri dish where the bacteria is growing and we are provided with a medium. And that medium has lactose. and in the medium there is no glucose available, then definitely these lactose has to enter into the cell of the bacteria. For that, permease is needed. And therefore, the expression of this lac operon at a very slower rate is always needed, because the lactose has to enter into the cell of the bacteria, and for that it needs to get permeated, and that permeability of the cell is increased by the presence of an enzyme, and that is called as permeases, right? Now, so here, by now we know that the lactose is acting as an inducer. Why it is acting as an inducer? Because the presence of the lactose is needed for the coming of the beta galactosidase, because if there is no lactose, we don't need beta galactosidase. The need of the beta galactosidase enzyme is present when only we can see, then only we can find it. That means lactose has to be present. So here the lactose then induces the operon in the following manner. We now said that lactose, it is acting as an inducer for lac operon. Why we have selected this lactose for inducing this process? Because the presence of lactose requires presence of beta galactosidase. If there is no lactose, we don't need beta galactosidase. That is what we consider this lactose as an inducer for the lac operon. Now, the repressor of the operon, this is a repressor of the operon is synthesized, and this will be no matter whether it is going to be proceed or it is not going to be transcribed, always repressor will be present. And this repressor, as it is presenting all the time, the presence of the repressor can be seen all the time. That is what we consider them as a all the time constitutively. And this type of the genes are called as constitutive genes. Constitutive genes are nothing but the genes which express all the time, no matter whether it is going to proceed or not going to proceed. The production of the repressor is seen all the time. So if you look at the operator, that is a lac operon, we find in the lac operon we have a promoter, we have a I gene which will transcribe and translate into repressor, we have the presence of promoter, we have the presence of operator, we have the presence of structural genes like Z, Y, A. So always the I gene will transcribe and translate to produce repressors. Repressors will be present all the time. This is very, very important, all the time. That is what we consider them as a constitutive gene. Constitutive genes are genes those express all the time. The repressor protein, this is the repressor protein, it is produced and it will go and bind to the operator region of the operon. Once it is produced, it will go and bind to the operator. This is operator region of lac operon. It will go and bind to the operator region. Then what happens? And prevent the RNA polymerases from transcribing the operon. We know that here is a promoter region, and to this region RNA polymerases enzyme come and attach, and it will proceed for the transcription of its structural gene. But here what happened? The repressor which is produced, it is going and attaching to the operator which is present next to the promoter, and RNA polymerases go and bind to the promoter. So here there is an obstruction. See, here is an operator region, and to the operator region repressor go and fix. So this is how we can see in the operon the repressor go and bind. Now this is the promoter region where RNA polymerases come and sits. This RNA polymerases find this as a obstacle, and this RNA is prevented for the further transcription to happen and further expression of the ZYA lac operon, which is going to produce beta galactosidase, permeases and transcriptase. This will not happen because it has been obstructed here. So the repressor binds to the operator region and prevents the RNA polymerases from transcribing the operon. If the lac mRNA is not transcribed, then there is no translation, there is no production of the enzymes. Beta galactosidase, permeases and transamylases are not produced. So now, this has happened. There are two cases. Repressors will all the time it will be available. So one case where repressor binds to operator region of lac operon. This is one condition. And other condition, repressor did not bind. Let's understand about the second, where the repressor is not going to bind to the operator region. In the presence of inducer, what is our inducer? Inducer is lactose or allolactose. Both are same, but the bonding is different. We can find the beta 1,4 bonding in the case of lactose and beta 1,6 bonding in the case of allolactose. But both are disaccharides which are going to break down to produce lactose and glucose, and the produced glucose will be acted as a source of energy. So in the presence of an inducer such as lactose or allolactose, the repressor is inactivated. If the lactose is not there, if the inducer is not there, the produced repressor is going to bind to the operator, right? But now this repressor is not free to go and bind to the operator. Before that happens itself, the repressor has been inactivated by binding with the inducer. Repressor is inactivated by interaction with inducer. So here we can see that this is a promoter region for the I gene. I gene, it is a constitutive gene. All the time it will be active, it will express. First it is transcribed into repressor mRNA, then it is translated into repressor protein. Now we have repressor protein. If there is no inducer, this repressor is going to bind to the operator region of the operon. But now in this case, this is the second case we are dealing with. We have lactose. This lactose will act as an inducer and go and bind to the repressor. Once the repressor is interacted with the inducer, this is inducer. Inducer is your lactose and this is repressor. This repressor, after binding with the inducer, it is going to be inactivated. It is going to be inactivated. Now the repressor will not bind to the operator. Repressor does not bind to the operator. Then where is the obstruction for the RNA polymerases to be get? Not to transcribe. It will happily transcribe now. Now here RNA polymerases enzyme, that is sigma factor, along with the sigma factor it will find the sequence of the promoter, then it will proceed towards the transcription of the gene which will produce lac mRNA, and further this is transcription and further it will translate into enzymes, and that enzymes are beta galactosidase, permeases, transacetylases. This allows the RNA polymerases get access to the promoter, and the transcription for the translation proceeds to produce proteins, that is enzymes. So essentially regulation of lac operon can also be visualized as a regulation of enzyme synthesis by its substrate, because Here the substrate is lactose. Lactose is acting as an inducer for the synthesis of its enzyme, and that enzyme is beta galactosidase. Beta galactosidase. Simple. So regulation of the lac operon by the repressor, which is going to bind to the operator, is refers to as a negative regulation. Even positive regulation is also exists for the lac operon, but here in this example, regulatory proteins can regulate both positively as activators or negatively as repressors. As the regulatory proteins in the case of lac operon are regulating by formation of repressor protein, we can take it as a repressor, and this is going to regulate negatively. We call it as a negative regulation. Negative regulation, because the regulatory protein, that is gene, are transcribed and translated into repressor, and this repressor protein are going to bind to the operator of the lac operon. That is what it shows, the negative regulation. That is it. So this is very clear now, where this is a promoter and I gene, where it will produce repressor mRNA translate into repressor. Here there is no inducer. In absence of inducer, there is no availability of the lactose. Then whatever produced repressor will go and bind to the operator which is present next to the promoter. Then there is an obstruction for this RNA polymerases which binds to the promoter and to proceed, because it is going to present as a abstract, and there is no, the repressor binds to the operator region and prevents the RNA polymerases from transcribing the operon. As there is no transcription, there is no translation, there is no production of the enzymes. And here in the second case, where the second case is the presence of inducer, and that presence of inducer is in the form of lactose. Same, this gene will express at all the time constitutive gene. Here also the I gene transcribe to produce repressor mRNA, and this repressor mRNA translate to produce repressors. Once the repressor is produced, it will get induced, it will get interacted with the inducer, and that inducer is lactose. Therefore, the repressor converts into inactive repressor. Now, this inactive repressor is unable to bind to the operator. Now, the promoter region is free, where RNA polymerases identify the sequence of the promoter, and it proceeds towards the transcription. Once the transcription is done, we have lac mRNA. Once the transcription is done, we have lac mRNA, and that will proceed towards translation to produce enzymes, which will be useful for the metabolism of the lactose. So that is it. So in this class, we had a very detailed description about how the lac operon will operate. In the coming up session, let's discuss about the human genome sequencing. Until then, keep learning with We Learning. Thank you.